Okay. Okay guys, in this video we will be talking about the immunology section, and in the subsequent videos also we'll be talking about the very important topic of immune system, which is called the T cell maturation and B cell maturation. So I'll be covering two videos. One is for T cell maturation, another one is for the B cell maturation. Then you will be seeing how this T cell maturation and B cell maturation, both of the pictures come to play along with each other. Okay, so before understanding the concept, now in this video we'll be talking about T cell maturation. Okay, so we'll be talking about T cell maturation. But before understanding any kind of T cell or B cell maturation, you must understand a very fundamental concept. And I assume before understanding and describing all the process that you must know the basics of immunology before coming to this section, because to understand this section you must have idea about what are B cells, what are T cells, what are their functions, and also what are called the MHC or major histocompatibility complex. If you don't have the idea of MHC complexes or about this B cells and T cells, I'll recommend you to go back to my YouTube channel and you can find videos about all these concepts. So just go back and look at those concepts, then come back and look at this video. Okay, so for this you must understand the fundamental concept of antigen processing inside the cell, because all of the T cell maturation as well as B cell maturation begins with the antigen processing. Now what are the antigens? Remember what we know about antigens. Antigens are the segment of foreign particles, right? Or it can be protein, it can be carbohydrate, it can be long polypeptide sequences, it can be lipids also. So whatever this thing is, it is of foreign origin, or it is non-self. Better write as non-self origin. So this thing is non-self. That means whatever is not present in us, if it is attacking us, we call it non-self or antigens in immune system. Okay, so whatever is invading into our house, we don't know them, our cell does not know them, we call them non-self or antigenic molecules. Now here says antigen molecules are coming inside our body. Now what they are doing, they are in our bloodstream. From the bloodstream they are coming to different regions. Now inside the blood, what we are having cells destined to fight against this kind of antigens. Because these are non-self, we need to take them and throw them out of our body. So for that there are cells inside our body. They are called different types of cells are there. One such type of cell is macrophage. One such cell is macrophage, and also there is cell like neutrophil. There is macrophage and there is neutrophil. Both of the functionality of the cells are taking up these antigens, kill them and chop them up and throw them away. But the functionality and the mechanism of taking these antigens and killing them is completely different of this macrophage and the neutrophil. So they are varying in this procedure, but the purpose of both the cells are similar or are same. Okay, now we must understand the procedure of engulfment of antigens and processing of the antigens by this type of cells. Both this type of cells are also called APC or antigen presenting cell. So we can also call them APCs. So both of them can be called as APCs or antigen presenting cells. So these cells are responsible for taking up antigens, processing the antigen and presenting the antigen. So taking, processing and presenting, right? So receiving, processing and presenting, these three are the important steps of the antigen processing by the macrophage as well as by the neutrophils. Okay, now what are the basic difference of this process? Macrophages, neutrophils, what neutrophil does, neutrophil take antigens, engulf antigens, and after engulfing the antigen, it will chop these antigens up and throw them away. Simply, it will chop them up and throw them away. So the action of neutrophil, say this is the neutrophil, it will engulf the antigen, say it engulfs the antigen, and after engulfing the antigen is here, say inside here, red color thing is the antigen. Say this thing is the antigen. After it taking the antigen, it will chop the antigen. It will chop the antigen into small segment, and then it throws it away. So then it throws it away, right? Just it cracks its cell and throws this antigen away. This is the function of neutrophil. I write it N for the neutrophil. Okay, so it's very basic. But the functionality of macrophage is little bit different than of neutrophil. What macrophage does, macrophage take up this antigens via specific kind of receptor. Okay, they take this via specific type of receptor. So this is our macrophage. Inside the macrophage, it engulfs the antigen. Here somewhere there is this antigen, which it can be peptides or whatever type of antigen. Then it processes the antigen or chop the antigen into small protein peptide fragments. So it will chop it into smaller fragments. Let's see here. It cleave it down into the smaller fragments. Now, after making the antigen breaking the antigen into smaller fragments, it produces another set of protein molecules, or it expresses a set of protein receptors. That receptor is going to bind with this type of protein or peptide fragments. So they also make the receptor which is going to bind with this fragmentalized antigen and hold this fragmentalized antigen and express it to the surface of this type of cells. So then they are also making this type of molecules, and what they will do, so this is the cell and these are those molecules coming out, these are those receptors coming out from this macrophage, and then the small peptides will be attached and presented onto the surface of the macrophage. This is macrophage. Okay, so these are the differences of antigen processing inside the cell. So neutrophil is simply taking it up, chop it up and throw it away. But macrophage taking it up, chop it up, and then it is having a receptor which will bind with the antigen and it will allow it to be presented onto the surface of macrophage. Now this type of molecules that is presenting the chopped out version of the antigen is called MHC molecules, or called major histocompatibility complex molecules. There are two types of major histocompatibility complex molecules. One is MHCI, another one is MHCII. These are of MHCII type of major histocompatibility complex molecule. Now what are the MHCI, MHCII, what are the difference? I will recommend you to go to my video in YouTube and you will find the answers. Okay, so this is the basic difference between the antigen processing by macrophage as well as by neutrophils. This idea is really important in future purpose. Now let us consider to our purpose T cell maturation. Now for the T cell maturation, this T cell or B cell, both of the cells, B lymphocyte and T lymphocyte, they are the part of acquired immune system, right? Because they are sitting there, so they must be activated somehow. How can they be activated? For the activation of T cell as well as B cell, we require some chemical mediators. From where the chemical mediators are coming? Only after the presenting and preparation and of these antigens so this type of antigen processing is important to throw the chemical signals to this T cell as well as B cells for them to be mature, right? So this is a very important part. So for the maturation of T cells, it is a role for the macrophage to give the signal. For the maturation of B cells, it is the role of neutrophils to give the signal. Remember this thing. So for the maturation of B cell, neutrophil gives the signal. For the maturation of T cell, macrophages provide the signal. Now in this case, for the maturation of T cell, we are going to see that macrophages are providing the signal to T cell that yes, it is the time for your maturation, for your expansion and propagation. And sooner, by this kind of chemical signals sent by this type of macrophages to T cells, it will activate T cell, and suddenly it will activate a pool of T cell. So lot of T cell similar type start to form inside our body to fight against this particular type of antigen which is just invading our tissue. Okay, so now let's see in this case how the T cell maturation is done. But again, going into that, another very important concept is that inside our body, what is going on, whatever type of maturation we are going to see, either T cell or B cell, a variety of cells, a variety of T cell and a variety of B cell always are prepared, and they are pre-programmed to have some important features onto them. They are pre-programmed, and the variations are provided from the beginning of our birth. The variations are provided. It doesn't mean that when an antigen enters, we modify a T cell or modify a B cell. Modification is done is a very little amount, but the actual variation was there from the beginning of the infection, even before of the infection. For example, suppose there are 4 T cells are there. Both of them are different. They are varying in their property, varying in their chemical nature. So one of them can be selected. This was the selected T cell, right? Suppose this one, 1, 2, 3 and 4, and this is the second one which is getting selected. So now this will expand. This will provide us the T cell pool after getting the signal from macrophage, right? So always the variation was there, and the variation which is created for T cell maturation as well as for B cell maturation are random in nature. So their random variations are there. How the random variations are formed? It's due to the gene shuffling. Gene shuffling is a very important part. So we need to start the understanding from gene shuffling to look at the variations that are there in T cell as well as in B cell. Now the gene shuffling means, for example, this is our T cell. So this is our cell. This is our T cell. Now this cell is consisting of chromosomes. Now the chromosome number 6 is a very important chromosome for T cells and for B cells also. Because in this chromosome number 6, say I'm going to draw the chromosome number 6, say this one and this one. So this is the chromosome number 6. Now in the chromosome number 6, there is a region in the short arm of the chromosome which are the region called the variable region of that gene. Now this variable segment of chromosome number 6 gene present in T cells are responsible for the gene shuffling. What it means, by shuffling of the genes in this region of the red colored region here, it can produce a variety of mRNAs. That means it can produce a variety of proteins from a single set of genes. Okay, so it can produce variety of proteins. Say it can produce a protein like round shape. It can make a protein like this. It can make a protein like this rectangular shape, or it can make a protein like this star shape. Okay, so it can make proteins like it wants from the single set of genes by simply varying this variable region sequence. Okay, by this nature, it can generate a random way of producing versatility among the pool of T lymphocytes that are present inside our body from the beginning of our life, and also before the antigen has entered into our body. So it is making all these things for a purpose. If antigen enters, it can go against it, and its effort definitely worthwhile. Now say it is varying its content, so what it will generate? A variety of, so it will generate sooner a variety of T cells there. Say 4 varieties, as we are dealing with 4 different types of variations. Let's say 4 different varieties of T cells are produced from the beginning. Okay, and they are responsible, and they are having different types of expression, protein expressions inside their body. But all of these T cells are having some important receptors common into their surface. There are some important receptors that must be present at their surface, which is really important here. Okay, So they are just ready for that. Okay. So what we know, antigens are coming, they are being processed by macrophage. How they are processed? Macrophage engulfs them, chew them up, then take them and attach them with MHCII molecules, hold them onto the surface. Till that part everything is fine. Okay. Now let's say in this case the antigen which enters into our body, it's a triangular antigen. Say the antigen is a triangular antigen. So here it processes the antigen and it is triangular in nature. So it's a triangular antigen what it gets. Okay. And the MHCII molecules of this macrophage is holding the triangular antigen onto the surface. Now what it will do, this macrophage will move from our bloodstream to the lymphatic system, from our bloodstream to the lymph nodes where the T cells are residing, and they are making these variations in their pool. So this macrophage will move there. So macrophage is coming there. Say here is our macrophage along with our MHC molecules holding that triangular shaped antigen. Okay. Now onto the surface of the T cells, as I have told you, due to this kind of mutation, due to this kind of changes or gene shuffling in their chromosome, they have prepared different type of T cells which are having or expressing different type of receptors onto their surface. So for example this one, as it is made by from this, so it is having the receptor for this circular shaped antigen. This one is having the receptor for this triangular shaped molecule. This one is having the receptor for this rectangular shaped antigen. This one is having the receptor for star shaped antigen. Okay. So now as we know, the antigen which is entered into our body is a triangular shape, and there is the pool of T lymphocytes that are generated by random shuffling of genes. Among these four, this second one is most suitable for the bonding with this kind of antigens which are being presented by the macrophage, right? So this one is the macrophage and these are all the T cells, right? So this is the interaction between macrophage, which is moving from the bloodstream to the lymphatic onto the lymph node, with the T cells which are sitting onto the lymph node, and the interaction will be selected on via the surface molecules. Now here the attachment and the interaction with this antigen is most suitable by this surface molecule of T2. So this type of T cells are going to bind with this type of triangular shaped antigen presented by the macrophage. So they will attach. After the attachment, so they will bind with each other. So let's zoom into the picture and draw it. So here is the T cell and here is the macrophage. So this macrophage is carrying the MHC molecule, and onto the surface of the MHC molecule it is providing this antigen, red colored antigen, triangular antigen. Now it is also having the triangular receptor, and with the help of this triangular receptor it is attached with this kind of antigen which is being processed by macrophage and then supplied to the T cell. So T cell here, so this is the T cell and this is the macrophage. They are interacting in the lymph node. Okay, and the first mode of interaction is between these MHCII molecules of macrophage and the surface receptor of T cell, which is called T cell receptor, because the receptor is present on the surface of T cell. So these are called T cell receptor or TCR, and we know this one MHCII, and we have already talked about that before. So first interaction between T cell and macrophage is via T cell receptor, and MHC2 molecules holding the antigen molecules. And from this population of T cells, the second one is selected because it is matching, the receptor of is matching, and it is complementary with the antigen which has just entered, right? So this is very important. So there are the clone of T cells are there, and one of them will be selected. This particular selection of T cell from a variety, from a pool of T cell, is called T cell selection or clonal selection, because there are a lot of clones of T cells that are present before the entry of antigen inside our body, inside the lymph nodes. And now one of those T cell is selected by macrophage. This is called clonal selection. So this procedure is called clonal selection. Okay. Now after this attachment, so this is the first attachment is done. But this attachment is not enough for the actual binding and further interaction of T and macrophage. So they want another signal to confirm the whether the antigen this T cell is receiving is particularly or perfectly modified, presented or not. Because T cells are very what you can say, picky about their selection of antigens. So not only the attachment via the TCR is giving them the signal, or not only the attachment can give them the satisfaction, but they want to make sure that these antigens are not only fitting onto the TCR, but also they are modified properly by the MHC2. So they also want to check whether the attachment of antigen with TCR is modified by the MHC2 or not. So whether there is a MHC2 present or not. So let me draw it a little more clearly, otherwise you can't visualize this. So let me draw here, say this is our macrophage, say, and say here is our MHC2. Okay, so this is MHC2. Okay, so for checking whether the antigen is properly processed by MHC2 or not, it provides another signal, okay, via another molecule that is present in T cell, which is called CD4. Now it provides the CD4. So this one is, I don't know whether you can visualize a green color or not. Let me mark it like that. So it provides another receptor which is called CD4. So this receptor is CD4. Using this CD4 receptor which is coming out from the T cell, it is attached with this MHC to check whether the antigen which is provided by the macrophage is properly processed with MHC2 or not. If it finds that this antigen is not properly processed, then the binding will be loosened out and this T cell will move away, a macrophage will move away. But now in this case, we know that this antigen is properly processed here by the MHC2 molecule, so they will stick to it. Okay. And for the further and final signaling procedure, it provides another important insight with the help of another set of interacting molecules that are present in T cell surface as well as in macrophage surface. And that molecule is called CD28, which is provided by, say, this one. So this is the CD28. Now what are these CDs? CD means cluster of designation or cluster of differentiation. When scientists start to research on these molecules and they found the molecules which are found in clusters and they are having similar type of properties, they mark them as cluster of designation. So similar type of molecules are in cluster of designation 1. Another set of similar cluster molecules which are having different property, so they mark them cluster of designation 2 or CD2 and so on. So this is the cluster designation number 28 molecule which is present in the T cell. And then there is another receptor for this CD28 onto the surface of macrophage, which is called B7. So these are the three interactions which are very important between the T cell and macrophage to finally provides some chemical signal which will tell T cell to grow and mature further. Okay, now in this case, first interaction is via TCR and MHCII molecule. Second is via the CD4 coming out from T cell to check whether the antigen is properly processed or not. And thirdly is via the CD28 coming from T and B7 coming from macrophage. After every successful attachment via this kind of receptors, it provides a signal to the inside of T cell. Okay, for example, say this is the nucleus of T cell. After each successful interaction of these surface receptors, it will provide the signal. So the first signal is coming from TCR and MHCII to the nucleus. Second signal via CD28, whether the properly modification of antigen is done or not, into the nucleus of T cell. Thirdly, the interaction between CD28 and B7 is again provided a strong signal to the nucleus of T cell. So from all the direction, nucleus of T cell is receiving the response, and this response is telling the nucleus for the gene expression of suitable genes which are required for the growth and maturation, right? Now there are chromosomes present inside the nucleus of T cell. Say let us draw the chromosomes like this red. So these are the chromosomes that are present inside the T cell. For example, from all the direction, signal is coming after the amplification of the signal inside the cell, and it will received by the chromosome and the genes. Now there are many genes that are present inside these chromosomes, and among those genes, 3 genes play a very important role in the maturation of T cell and B cell. Now in this case, 2 genes play a very vital role in the maturation of T cell. Okay, so suppose this is the gene number 1 here, and say this is the gene number 2 here. Gene number 1 and gene number 2 residing there. Now gene number 1, when it is getting amplified, when it is receiving the signal and it is expressing itself, it will start to produce and secrete some molecules outside this T cell. So they will start providing some molecules outside this cell. Say these are the molecules. These molecules are called interleukin 1. They are called interleukin 1. Okay, not 1, sorry, they are called interleukin 2. Mistake. They are called interleukin 2. Okay, so they are receiving the signal, and after receiving the signal start producing interleukin 2 molecules. Now these molecules are going to interact with someone with whom they are interacting in this case. Now here comes the role of gene 2. Gene 2 is also producing another component. This component is also a protein component which is helping to provide a receptor onto the surface of this T cell. So it is providing the receptor onto the surface of T cell. So gene 1 is responsible for providing the molecules and excreting the molecules which are called interleukin 2, and gene 2 is providing and making the receptor for the interleukin 2 onto the surface. Now what will happen? These molecules will come and sit onto this receptor region. So these interleukins are secreted by T cells because of the expression of gene 1 and are attached to this kind of surface receptors which are provided by gene 2. So in this case, this T cell is secreting something which is indeed expressing itself, which is indeed acting on itself. This type of action is called autocrine action. So I am providing something, and that is in turn is acting on me. It is called the autocrine action. Auto means itself. So it's an autocrine activation is done. Due to this kind of activation, it gives the cell a signal to grow and divide rapidly. So it will eventually provide the final signal to this T cell, and the signal is mitosis, growth and division. So by receiving the signals of mitosis, sooner this type of T cell will start to form a pool of T cells, right? So they start to form. So let me make it smaller. So yes, so these are the pool of the T cell, similar type of T cell. All of the T cells are having particular T cell receptors, exact T cell receptors to receive this type of delta shaped antigenic molecules. So they will start to propagate and produce a pool. Now this particular part, when they get the signal via interleukin 2 and they get expanded, right? Because from the very beginning there are a particular pool which is getting selected, so we call it a clonal selection. But now from that type of clone which is selected, they are getting amplified due to this kind of growth and mitotic signal they are receiving by the interaction of macrophage. And this type of signal, after receiving this signal, they are triggered to produce more for the expansion. So this particular step is called clonal expansion. Okay, so clonal expansion or clonal propagation, because we are getting many clones now of the same time. So we are getting clonal expansion in this case. So it is called, so let me write it here. It is called clonal expansion. I hope you can see it. It is called clonal expansion. So very beginning it's clonal selection, then clonal expansion of T cell is done, and after clonal expansion we can tell that this T cell is getting matured now. Now from the very beginning the T cell was not matured. The T cell we are dealing with at the very early, this is called, those are called the naive, and then they are modified to produce these cells. They are called TH cells or helper T cells, right? Now these cells are getting modified due to many chemical signaling procedure by the interaction with macrophages. So here these macrophages, again I am telling you, they are the teacher of these T lymphocytes. And on the other hand, I have also told you from the very beginning of our discussion that neutrophils are the teacher of B lymphocytes and macrophages are the teacher of T lymphocytes, which we have seen. Now we'll be seeing the maturation of B cells, right?